Let's continue with the renal physiology. In this video, we are going to learn about urine formation by the kidneys, renal blood flow, glomerular filtration, and their control. So, some learning objectives are included like describe peculiarities of renal blood supply and blood flow, describe the process of glomerular filtration and its measurement, describe the regulation of glomerular filtration, describe the structure of the juxtaglomerular apparatus. Some important terms are associated with this, like colloid osmotic pressure, also called oncotic pressure, is a form of osmotic pressure exerted by proteins in the blood plasma. Water tends to flow into the plasma as a result of this pressure. Then capillary filtration coefficient, or Kf, a factor that is representative of the permeability of the capillary wall and its area. Then prostaglandins, group of lipid-derived compounds that have a variety of local autocrine or paracrine physiological effects, such as regulating the contraction and relaxation of smooth muscle tissue. Then bradykinin, peptide derived from a plasma precursor causing vasodilation. Urine formation results from globular filtration, tubular reabsorption, and tubular secretion. The rates at which different substances are excreted in the urine represent the sum of three renal processes: glomerular filtration, reabsorption of substances from the renal tubules into the blood, secretion of substances from the blood into the renal tubule. Then expressed mathematically, all these factors we can just write like urinary excretion rate is equal to filtration rate minus reabsorption rate plus secretion rate. So filtration will be happening. So its rate is there. From that we need to subtract the reabsorption rate. Reabsorption is something which is happening, which decreases the urine produced, which is excreted. So along with that we will be adding the secretion rate. So urine formation begins when a large amount of fluid that is virtually free of protein is filtered from the glomerular capillaries into Bowman's capsule. Most substances in the plasma except for proteins are actually filtered. Most substances in the plasma except for proteins are freely filtered, so their concentration in the glomerular filtrate in Bowman's capsule is almost the same as in the plasma. As filtered fluid leaves Bowman's capsule and passes through the tubules, it is modified by reabsorption of water and specific solutes back into the blood or by secretion of other substances from the peritubular capillaries into the tubules. As filtered fluid leaves Bowman's capsule and passes through the tubules, it is modified by reabsorption of water and specific solutes back into the blood or by secretion of other substances from the peritubular capillaries into the tubules. Figure 77 shows the renal handling of four hypothetical substances. The substances shown in panel A is freely filtered by the glomerular capillaries but is neither reabsorbed nor secreted. Furthermore, its excretion rate is equal to the rate at which it was filtered. Certain waste products in the body, such as creatinine, are handled by the kidneys in this manner, allowing excretion of especially all that is filtered. In panel B, the substance is freely filtered, but it also partly reabsorbed from the tubules back into the blood. Therefore, the rate of urinary excretion is less than the rate of filtration at the glomerular capillaries. In this case, the excretion rate is calculated as the filtration rate minus the reabsorption rate. This pattern is typically for many of the electrolytes of the body, as a sodium and chloride ions. In panel C, the substance is freely filtered at the glomerular capillaries, but it is not excreted into the urine because all the filtered substances is reabsorbed from the tubules back into the blood. This pattern occurs for some of the nutritional substances in the blood, such as amino acids and glucose, allowing them to be conserved in the body fluids. The substances in panel D is freely filtered at the glomerular capillaries and is not reabsorbed, but additional quantities of the substance are secreted from the peritubular cavity blood into the renal tubule. This pattern often occurs for organic acids and bases, permitting them to be rapidly cleared from the blood and excreted in large amounts in the urine. The excretion rate in this case is calculated as filtration rate plus tubular secretion rate. For each substance in the plasma, a particular combination of filtration, reabsorption, and secretion occurs. The rate at which the substance is excreted in the urine depends on the relative rates of these three basic renal processes. Why are large amounts of solutes filtered and then reabsorbed by the kidneys? One might question the wisdom of filtering such large amounts of water and solutes and then reabsorbing most of the substances. The advantage of high glomerular filtration rate is that it allows the kidneys to rapidly remove waste products from the body that depend mainly on glomerular filtration for their excretion. Most waste products are poorly reabsorbed by the tubules and therefore depend on a high GFR for effective removal from the body. A second advantage of a high GFR is that it allows all the body fluids to be filtered and processed by the kidneys many times each day, because the entire plasma volume is only about 3 liter, whereas the GFR is about 180 liter per day. The entire plasma can be filtered and processed about 60 times each day. This high GFR allows the kidneys to precisely and rapidly control the volume and composition of the body fluids. And you just try to learn the basic kidney process that determine the composition of the urine. Urinary secretion rate of a substance is equal to the rate at which the substance is filtered minus its reabsorption rate plus the rate at which it is secreted from the peritubular capillary blood into the tubules. So afferent arteriole and efferent arteriole is there, glomerular capillaries will be there, filtration, reabsorption, secretion and excretion will be happening, and Bowman's capsule is there. So peritubular capillaries and renal vein and urinary excretion can be seen. So excretion is equal to filtration minus reabsorption plus secretion. So A, filtration only happening substance. Filtration and partial reabsorption is happening in B, substance B. Then filtration, complete reabsorption is happening in substance C, and filtration and secretion is happening in substance D. Renal handling of four hypothetical substances. A, the substance is freely filtered but not reabsorbed. B, the substance is freely filtered but part of the filtered load is reabsorbed back in the blood. The substance is freely filtered but it's not excreted in the urine because all the filtered substances is reabsorbed from the tubule into the blood. The substance is freely filtered and is not reabsorbed but is secreted from the peritubular capillary blood into the renal tubule. So, renal blood flow is another topic. Renal blood supply. Blood flow to the two kidneys is normally about 22% of the cardiac output or 1100 ml per minute. The renal artery enters the kidney through the hilum and then branches progressively to form the interlobar arteries, arcuate arteries, interlobular arteries, also called radial arteries, and afferent arterioles, which lead to the glomerular capillaries, where large amounts of fluid and solutes, except the plasma proteins, are filtered to begin urine formation. The distal ends of the capillaries of each glomerulus coils to form an efferent arteriole, which leads to the second capillary network and the peritubular capillaries that surround the renal tubule. The renal circulation is unique in having two capillary beds, the glomerular and peritubular capillaries, which are arranged in series and separated by the efferent arterioles, which help regulate the hydrostatic pressure on both sets of capillaries. High hydrostatic pressure in the glomerular capillaries, about 6 mm per Hg, causes rapid fluid filtration, whereas a much lower hydrostatic pressure in the peritubular capillaries, about 13 mm Hg, permits rapid fluid reabsorption. By adjusting the resistance of afferent and efferent arterioles, the kidneys can regulate the hydrostatic pressure on both the glomerular and the peritubular capillaries, thereby changing the rate of glomerular filtration, tubular reabsorption, or both in response to body homeostatic demands. The peritubular capillaries empty into the vessels of the venous system, which run parallel to the arteriolar vessels. The blood vessels in the venous system progressively form the interlobular vein, arcuate vein, interlobar vein, and renal vein, which leave the kidney beside the renal artery and ureter. In an average 70 kg man, the combined blood flow through both kidneys is about 1100 ml per minute, or about 22% of the cardiac output. Considering that the two kidneys constitute only about 0.4 of the total body weight, one can readily see that they conceive an extremely high blood flow compared with other organs. As with other tissues, blood flow supplies the kidneys with nutrients and remove waste products. However, the high flow of the kidneys greatly exceeds this need. The purpose of the additional flow is to supply enough plasma for the high rates of glomerular filtration that are necessary for precise regulation of body fluid volumes and solute concentrations. As might be expected, the mechanisms that regulate renal blood flow are closely linked to the control of GFR and the excretory functions of the kidneys. Renal blood flow and oxygen consumption. On a per gram weight basis, the kidneys normally consume oxygen at twice the rate of the brain, but have almost 7 times the blood flow of the brain. Thus, the oxygen delivered to the kidneys far exceeds their metabolic needs, and the arterial venous extraction of oxygen is relatively low compared with that of most other tissues. So, when we just try to look at a picture of oxygen, it's a graph. Like oxygen consumption is on the x-axis, then sodium reabsorption on the y-axis, basal oxygen consumption is there, and through that 0.5 portion there is a straight line. So relationship between oxygen consumption and sodium reabsorption in dog kidneys, the straight line can be seen. A large fraction of the oxygen consumed by the kidneys is related to the high rate of active sodium reabsorption by the renal tubules. If renal blood flow and GFR are reduced and less sodium is filtered, less sodium is reabsorbed and less oxygen is consumed. Therefore, renal oxygen consumption varies in proportion to renal tubular sodium reabsorption, which in turn is closely related to GFR and the rate of sodium filtered. If glomerular filtration completely ceases, renal sodium reabsorption also ceases and oxygen consumption decreases to about 1/4 normal. This residual oxygen consumption reflects the basic metabolic needs of the renal cells. Then, determinants of renal blood flow. Renal blood flow is determined by the pressure gradient across the renal vasculature. The difference between renal artery and renal vein hydrostatic pressures divided by the total renal vascular resistance. Then renal artery pressure minus renal vein pressure by total renal vascular resistance. Renal artery pressure is about equal to systemic arterial pressure, and renal vein pressure averages about 3 to 4 mmHg under most conditions. As in other vascular beds, the total vascular resistance through the kidneys is determined by the sum of the resistance in the individual vasculature segments, including the arteries, arterioles, capillaries, and veins. Renal artery pressure is about equal to Systemic arterial pressure and renal vein pressure averages about 3 to 4 mmHg under most conditions. As in other vascular beds, the total vascular resistance through the kidneys is determined by the sum of the resistance in the individual vasculature segments, including the arteries, arterioles, capillaries, and veins. Most of the renal vascular resistance resides in three major segments: interlobular arteries, afferent arterioles, and afferent arterioles. Resistance of these vessels is controlled by the sympathetic nerve system, various hormones, and local internal renal control mechanisms. An increase in the resistance of any of the vascular segments of the kidneys tend to reduce the renal blood pressure flow, whereas a decrease in vascular resistance increases renal blood flow if renal artery and renal vein pressures remain constant. Although changes in arterial blood pressure have some influence on renal blood flow, the kidneys have effective mechanisms for maintaining renal blood flow and GFR relatively constant over an arterial pressure range between 80 and 170 mmHg, a process called autoregulation. This capacity for autoregulation occurs through mechanisms that are completely intrinsic to the kidneys. Blood flow in the vasa recta of the renal medulla is very low compared with flow in the renal cortex. The outer part of the kidney, the renal cortex, receives most of the kidney's blood flow. Blood flow in the renal medulla accounts for only 1 to 2% of the total renal blood flow. Flow to the renal medulla is supplied by a specialized portion of the peritubular capillary system called the vasa recta. These vessels descend into the medulla in parallel with the loops of Henle and then loop back along with the loops of Henle and return to the cortex before emptying into the venous system. The vasa recta play an important role in allowing the kidneys to form concentrated urine. Then autoregulation of GFR and renal blood flow. Feedback mechanisms intrinsic to the kidneys normally keep the renal blood flow and GFR relatively constant, despite marked changes in arterial blood pressure. This mechanism still function in blood perfused kidneys that have been removed from the body, independent of systemic influence. This relative constancy of GFR and renal blood flow is referred to as autoregulation. The primary function of blood flow autoregulation in most tissues other than the kidney is to maintain the delivery of oxygen and nutrients in a normal level and to remove the waste products of metabolism despite changes in the arterial pressure. In the kidneys, the normal blood flow is much higher than that required for these functions. The major function of autoregulation in the kidneys is to maintain a relatively constant GFR and to allow precise control of renal excretion of water and solutes. The GFR normally remains relatively constant despite considerable arterial pressure fluctuations that occur during a person's usual activities. For instance, a decrease in arterial pressure to as low as 70 to 75 mmHg or an increase to as high as 160 to 180 mmHg usually changes the GFR less than 10%. In general, renal blood flow is autoregulated in parallel with GFR, but GFR is more effectively autoregulated under certain conditions. Then myogenic autoregulation of renal blood flow and GFR. Another mechanism that contributes to the maintenance of a relatively constant renal blood flow and GFR is the ability of individual blood vessels to resist stretching during increased arterial pressure, a phenomenon referred to as the myogenic mechanism. Studies of individual blood vessels, especially small arterioles, throughout the body have shown that they respond to increased wall tension or wall stretch by contraction of the vascular smooth muscle. Stretch of the vascular wall allows increased movement of calcium ions from the extracellular fluid into the vascular smooth muscle cells, causing them to contract by combining with intracellular calmodulin and the activation of myosin cross bridges. This contraction prevents excessive stretch of the vessel and at the same time, by raising vascular resistance, helps prevent excessive increase in renal blood flow and GFR when arterial pressure increases. Although the myogenic mechanism probably operates in most arterioles throughout the body, Its importance in renal blood flow and GFR autoregulation has been questioned by some physiologists because its pressure sensitive mechanism has no means of directly detecting changes in renal blood flow or GFR per second. On the other hand, this mechanism may be more important in protecting the kidney from hypertension induced injury. In response to sudden increase in blood pressure, the myogenic constrictor response in afferent arteriole occurs within seconds and therefore attenuates transmission of increased arterial pressure to the glomerular capillaries. Then glomerular filtration is there. So glomerular filtration, the first step of urine formation. Composition of the glomerular filtrate. Urine formation begins with filtration of large amounts of fluid through the glomerular capillaries into Bowman's capsule. Like most capillaries, the glomerular capillaries are relatively impermeable to proteins, so the filtered fluid called the glomerular filtrate is essentially protein-free and devoid of cellular elements including red blood cells. The concentration of other constituents of the glomerular filtrate, including most salts and organic molecules, are similar to the concentration in the plasma. Exceptions to this generalization include a few low molecular weight substances such as calcium and fatty acids that are not freely filtered because they are partially bound to the plasma proteins. GFR is about 20% of the renal plasma flow. The GFR is determined by the balance of hydrostatic and colloid osmotic forces acting across the capillary membrane. The capillary filtration coefficient, the product of the permeability and filtering surface area of the capillaries. The glomerular capillaries have a much higher rate of filtration than most other capillaries because a high glomerular hydrostatic pressure and a large KF in the average adult human. The GFR is about 125 ml per minute for or about 180 liter per day. The fraction of the renal plasma flow that is filtered, the filtration fraction averages about 0.2. This means that about 20% of the plasma flowing through the kidney is filtered through the glomerular capillaries. The filtration fraction is calculated as follows. Filtration fraction is equal to GFR by renal plasma flow. Then glomerular capillary membrane. The glomerular capillary membrane is similar to that of other capillaries except that it has three instead of the usual two major layers. The endothelium of the capillary, a basement membrane, a layer of epithelial cells, podocytes, surrounded by the outer surface of the capillary basement membrane. Together, these layers make up the filtration barrier, which, despite the three layers, filters several hundred times as much water and solutes as the usual capillary membrane. Even in this high rate of filtration, the glomerular capillary membrane normally prevents the filtration of plasma proteins. The high filtration rate across the glomerular capillary membrane is due partly to its special characteristics. The capillary endothelium is perforated by thousands of small holes called fenestrae, similar to the fenestrated capillaries found in the liver. Although smaller than the fenestrate of the liver, although the fenestrations are relatively large, endothelial cell proteins are richly endowed and fixed negative charges that hinder the passage of plasma proteins. Surrounding the endothelium is the basement membrane, which consists of a meshwork of collagen and proteoglycan fibrillae that have large space through which large amounts of water and small solutes can filter. The basement membrane effectively prevents filtration of plasma protein in part because of strong negative electrical charges associated with the proteoglycans. And just we try to look at the picture which represent the basic in ultrastructure of a glomerular capillary. You can see that there are proximal tubule, podocytes, capillary loops, Bowman's space, Bowman's capsule, and afferent arteriole. And we just try to look at the picture of cross section of the glomerular capillary membrane and its major components. Capillary epithelium, basement membrane, and epithelium or podocytes can be seen. Slit pores will be there. Epithelium is there, basement membrane is there, endothelium and fenestrations can be seen. The final part of the glomerular membrane is a layer of epithelial cells that line the outer surface of the glomerulus. The cells are not continuous but have long foot-like processes or podocytes that encircle the outer surface of the capillaries. The foot processes are separated by gaps called slit pores through which the glomerular filtrate moves. The epithelial cells, which also have negative charges, provide additional restriction to filtration of plasma proteins. Thus, all layers of the glomerular capillary wall provide a barrier to filtration of plasma proteins. Filterability of solutes is inversely related to their size. The glomerular capillary membrane is thicker than most other capillaries, but it is also much more porous and therefore filters fluid at a high rate. Despite the high filtration rate, the glomerular filtration barrier is selective in determining which molecule will filter based on their size and electrical charge. The filtrability of 1.0 means that the substance is filtered as freely as water. A filtrability of 0.75 means that the substance is filtered only 75% as rapidly as water. Note that electrolytes such as sodium and small organic compounds such as glucose are freely filtered. As the molecular weight of the molecule approaches that of albumin, the filtrability rapidly decreases, approaching zero. Negatively charged large molecules are filtered less easily than positively charged molecules of equal molecular size. The molecular diameter of the plasma protein albumin is only about 6 nm, whereas the pores of the glomerular membrane are thought to be about 8 nm. Albumin is restricted from filtration, however, because of its negative charge and electrostatic repulsion excreted by negative charges of the glomerular capillary wall proteoglycans. Electrical charge affects the filtration of different molecular weight dextrins by the glomerulus. Dextrins are polysaccharides that can be manufactured as neutral molecules or with negative or positive charges. Note that for any given molecular radius, positively charged molecules are filtered much more readily than are negatively charged molecules. Neutral dextrins can also filter more readily than are negatively charged dextrins of equal molecular weight. The reason for this difference in flexibility is that the negative charge of the basement membrane and the podocytes provide an important means for restricting large negatively charged molecules, including the blood plasma proteins. In certain kidney diseases, the negative charges on the basement membrane are lost even before this are noticeably changes in kidneys histology, a condition referred to as minimal change nephropathy. As a result of this loss of negative charge on the basement membranes, some of the lower molecular weight proteins, especially albumin, are filtered and appear in the urine, a condition known as proteinuria or albuminuria. Next topic, use of clearance methods to quantify kidney function, GFR and RBF. The rates at which different substances are cleared from the plasma provide a useful way of quantitating the effectiveness with which the kidneys excrete various substances. So, by definition, the renal clearance of a substance is the volume of plasma that is completely cleared of the substance by the kidneys per unit time. This concept is somewhat abstract because there is no simple volume of plasma that is completely cleared of a substance. However, renal clearance provides a useful way of quantifying the excretory function of the kidneys, and as discussed later, can be used to quantify the rate at which renal blood flows to the kidneys, as well as the basic functions of the kidneys, glomerular filtration, tubular reabsorption and tubular secretion. To illustrate the clearance principle, consider the following example. If the plasma passing through the kidneys contains 1 mg of a substance in each milliliter, and if 1 mg of the substance is also excreted into the urine each minute, then 1 ml per minute of the plasma is cleared of the membrane substance. This clearance refers to the volume of plasma that would be necessary to supply the amount of substance excreted in the urine per unit volume time. Stated mathematically, Cs into Ps is equal to Us into UV. Since Cs is the clearance rate of a substance, Ps is the plasma concentration of the substance, then Us is the urine concentration of the substance, and V is the urine flow rate. Rearranging this equation, clearance can be expressed as Cs is equal to Us into V by Ps. Thus, renal clearance of a substance is calculated from the urinary excretion rate US into V of that substance divided by its plasma concentrations. So, insulin clearance can be used to estimate GFR. If a substance is freely filtered as freely as water, and is not reabsorbed or secreted by the renal tubules, then the rate at which that substance is excreted in the urine, US into V, is equal to the filtration rate of the substance by the kidneys, like GFR into PS. Thus, GFR into PS is equal to US into V. The GFR therefore can be calculated as the clearance of the substance as follows. GFR is equal to US into V by PS is equal to CS. A substance that fits this criteria is inulin, a polysaccharide molecule with a molecular weight of about 5200. Inulin, which is not produced in the body, is formed by the roots of certain plants and must be administered intravenously to a patient to measure GFR. Renal handling of inulin. In this example, the plasma concentration is 1 mg per ml, urine concentration is 125 mg per ml, and urine flow rate is 1 ml per minute. Therefore, 125 mg per minute of inulin passes into the urine. Then, inulin clearance is calculated as the urine concentration excretion rate of inulin divided by the plasma concentration, which yields a value of 125 ml per minute. Thus, 125 ml of plasma flowing through the kidneys must be filtered to deliver the inulin that appears in the urine. Inulin is not only the substance that can be used for determining GFR. Another substance that has been used clinically to estimate GFR include radioactive iothalamate and creatinine.